Okay, we'll go ahead and get started. Thanks everybody who's able to join us in person and on Zoom. It's my pleasure to introduce Ryan Chiu for his master's thesis defense today. Just a little bit of an introduction for Ryan. Ryan graduated from UCLA with a bachelor degree of science in atmospheric, oceanic, and environmental sciences with a minor in environmental systems and society. And building on this undergraduate degree, he gained a lot of relevant experience before coming to join our program at Moss Landing Marine Labs. So he worked at the UCLA Marine Operations Program, leading cruises as a boat operator and working with instrumentation. He continued developing his instrumentation expertise as a test engineer at the Google campus in Sunnyvale, and then went on to work at the U.S. Green Chamber of Commerce, working on a professional climate change certification course. So when Ryan came to the Physical Oceanography Lab in Moss Landing, he was well prepared to take on a number of different roles. He became a highly valued team member at the Moss Landing Marine Operations Department. He served as student body rep for SJSU. And he worked with the Central Coast Wetlands Group extensively on modeling and data analysis to support management in Moro Cojo Slough, which is the system he'll be talking about today. So that work with Central Coast Wetlands Group provided the foundation for his thesis project. And he also was an active member of the Moro Cojo Slough Technical Advisory Committee and is co-author on the Moro Cojo Slough Restoration and Enhancement Plan, which is currently in review. So now Ryan already has one foot out the door, and he's continuing on to the next step of his career as a scientist. So he's entered the PhD program at the University of Washington Civil and Environmental Engineering Department in Seattle. There he's working on the Cascadia Coastlines and Peoples Hazard Research Hub, which is working to prepare and adapt coastal communities in the Pacific Northwest of climate change. So a great extension of the work that he's been doing here in the PhysOcean Lab and at CCWG. And he's already working on numerical modeling of flooding in different estuaries in the Pacific Northwest. So it's my pleasure to introduce Ryan Chiu as he presents his thesis work, Spatiotemporal Dynamics and Biogeochemical Responses in a Tidally Restricted Moro Cojo Slough. Let's welcome Ryan.
Great. Thanks for that kind introduction, Tom. So thank you all for being here today. Today, I'll be presenting my work on spatiotemporal dynamics and biogeochemical responses in a tidally restricted Moro Cojo Slough. So what is an estuary? Estuary is a body of water that is located in the region where a river meets the salty open sea, and this could range from coastal marshes to bays and their surrounding wetlands. Now a slough, as you all might be familiar with, Elkhorn Slough is a type of wetland usually at the backwater parts of bays, but this still resides where the freshwater meets the open ocean. But for this presentation, I'll be using the terms estuary and slough interchangeably. So wetlands are extremely important interfaces as it plays a role in providing ecosystem services such as protection for coastlines and coastal communities. It does an amazing job at mitigating coastal erosion and flooding. It provides habitats for both terrestrial and marine organisms, and it also acts as water filtration for removing pollutants and contaminants from surface runoff, which can be extremely useful for especially urbanized and agricultural areas. Now, there are many types of estuaries, but today I'll be focusing on a Mediterranean climate estuary. Now, these unique estuaries can be found across multiple continents, from the Mediterranean basin in Southern Europe, South Africa, across central Chile and South America, California and the Baja Peninsula in North America, and parts of Southern Australia. And these can be characterized by two seasons throughout the year, either wet or dry. And for wet seasons, they follow a typical classic estuarine circulation with a seaward flow of typically fresh or less dense water at the surface, and landward flow at the bottom, typically comprised of salty seawater. During the dry seasons, however, that flow diverges from this pattern and can be characterized as an inverse circulation, where the lack of rainfall and dry climate drives high evaporation rates that greatly exceeds the amount of freshwater input or precipitation into the system. And this increase in density results in an exchange opposite to the wet season, where the ocean water actually flows into the slough at the surface, and the upstream slough water flows seaward at the bottom. And these conditions end up with a hypersaline environment upstream, where the reverse in circulation ends up dampening the exchange flow due to the weakened density gradient. And that lack of exchange and flushing actually increases the residence time of waters upstream, where they're left to evaporate and become even more saline. And many of these results, or many of these systems, have developed resilience and adapted to these conditions over time. But with the increase of anthropogenic factors over the past few centuries, these habitats have started to shift and degrade as a result. Now, historically, over the past 200 years, the US lost around 53% of its wetlands. Majority of this is due to land use change for agricultural, commercial, or residential uses, with agricultural being the most common. And here, especially in California, over the last 150 years, there's been a 91% loss in wetland area. And other factors that contribute to wetland loss is anthropogenically driven eutrophication from excessive nutrient loading, which is typically observed due to agriculture runoff. And if the system lacks strong tidal exchange, those waters may become more stagnant and become hypoxic if they're not flushed out routinely. Now, one way to measure the health of an ecosystem is by calculating the net ecosystem metabolism and using this as a metric to characterize the metabolic behavior of the system. So this can be thought of as a difference between gross primary production and ecosystem respiration. So when primary productivity, or the fixing of organic carbon, is higher than the fixing of inorganic carbon during respiration, the system can be characterized as net autotrophic and acts as a source for organic carbon. One example of this is the upper ocean, which acts as a net exporter or source of organic carbon to the deep ocean. And this mainly occurs in the euphotic zone, where there's a high rate of photosynthesis, and this results in a high rate of carbon fixing, and all of this organic matter eventually sinks down into the deeper ocean. In the opposite case, where respiration rates are higher than productivity, system can be categorized as net heterotrophic, which acts as a sink for organic carbon. And this is what estuaries are typically thought of, generally acting as a net sink for organic carbon, and as a result, it's typically characterized as net heterotrophic. And in some occasions, the system can be well balanced when both gross primary productivity and ecosystem respiration are nearly equal to each other. Now, for this study, I chose the Mediterranean climate Moro Cojo Slough as my study site for a couple of different reasons. For one, it's similar to its well-studied neighbor Elkhorn Slough, which also has a long history of anthropogenic modifications such as diking, grazing, and farming. Next, its vicinity to agricultural lands make it susceptible to high nutrient loading from agricultural runoff, which is widely known to contain large amounts of nitrate and phosphate. And so with enough of this concentration built up in a slough, this can eventually lead to eutrophication. And this figure on the top over here was from a study done by Hughes et al. And shows the eutrophication intensity throughout the region. And Moro Cojo Slough can be seen as hypereutrophic compared to its neighboring Elkhorn Slough, where more of the upper reaches in the slough are categorized as hypereutrophic. In addition to that, a one-way tidal restriction, seen as the black dot on the west side of that zoomed-in map, was installed over 3 decades ago at its mouth to prevent seawater inflow and also preserve upstream habitats. But as a result of this modification, other issues were introduced or exacerbated by limiting the tidal exchange, which then leads to more frequent hypersaline waters upstream. And this, combined with high nutrient loading from the agricultural fields, can further degrade the water quality of the slough. And this is well documented through the Elkhorn Slough report card, where it considers a range of parameters to determine a grade based on overall water quality of the estuary. And these records date back to 1990. And since then, Moro Cojo has received 27 F grades and 3 D grades. And even though there are long-term observations for Moro Cojo, it's been primarily used for monitoring purposes, and it's still a pretty understudied site, especially when comparing it to its neighboring Elkhorn Slough. There have been previous studies, such as a thesis done by Mo Wise here at Moss Landing Marine Labs, that looked at nutrient dynamics within a slough. and also a handful of other papers that briefly touch on the slough, but none of them have quantitatively looked at the individual factors that contribute to its eutrophic, hypersaline, and hypoxic conditions that's observed in these long-term observations. So tidal restrictions are commonly used and installed for many different reasons, such as for agricultural practices or mitigating upstream seawater advection. In this case, in Moro Cojo, it restricts flow in, but it also allows water to flow out based on a water level difference between a system and a harbor on the other side. So say in the cases where the water level in a slough is lower or equal to the harbor's water level, there won't be any inflow. But whenever water level is higher in the slough, that pressure difference will actually open the tide gate and allow water to naturally flow out. But with the tide gate installed, it also comes with impacts to the system, such as limiting the tidal exchange at the mouth, which leaves the system prone to the upstream regions drying up and becoming hypersaline, especially during the dry seasons. And a lack of replenishing the system with DO-rich waters allow the upstream regions to become hypoxic more frequently. And with the flow altered by the tide gate, it also limits the amount of organic carbon that can be exported through natural tidal exchange, and that ends up recirculating and getting stored in the slough. Lastly, it's important to note that the tide gate was installed at the mouth, failed back in 2016, but it was repaired. But even after it was repaired, there's still leaking around the edges that allow seawater to enter during flood tides. And now there aren't too many studies that hone in on describing net ecosystem metabolism and carbon budgets of estuaries as a result of these tidal restrictions. So that's another reason why I thought it would be extremely useful to look at this particular system. My thesis, I worked to answer two main questions. Number one, how do salinity, nitrate, and DO vary in response to tides, atmospheric forcing, and large precipitation events? And here I'll investigate how these patterns respond on a seasonal, fortnightly, and diurnal scale. Next, I look at how Moro Cojo's net ecosystem metabolism is characterized on a seasonal scale. And this basically means looking at whether activities related to either primary production or respiration dominates throughout the season. Thinking about the carbon budget, this can tell us whether the system typically acts as a net sink or source of carbon throughout the year. And ultimately, I'll be looking at whether a system follows the characteristic net heterotrophic pattern observed in most other estuaries in the US. And to answer these questions, I'll be looking at a time series over 4 years from 2017 to 2021 to observe any spatial or temporal patterns within a system. I separated the year into 2 seasons, with the dry season being from May to October and a wet season from November to April. And first, I'll be using data from monthly transects collected by the Central Coast Wetlands Group. Next, I'll be looking at data from continuously monitoring station located in Moro Cojo, Elkhorn Slough, and Monterey Bay. And next, I also deployed sensors to target spatial resolution and temporal variability of DO in Moro Cojo for each season. And lastly, I constructed box models or budgets to calculate salinity fluxes and DO fluxes to look at the major terms that ultimately contribute to the overall storage at the end of each season. And on a map here, we are all of these stations plotted, starting with the 13 stations that were sampled monthly by CCWG, where they collected measurements of water depth, temperature, salinity, DO, and perform nitrate grabs. And those can be seen in the colorful dots from the red hues at the mouth moving eastward up until, the blue hues at the mouth and then moving upstream to red hues all the way towards the North Monterey County High School. Next, the black dots here show the locations of each targeted deployment, which covered 4 sites along the slough. Next, there's the Moro Cojo Slough monitoring station located right underneath the Highway 1 bridge, and it uses a bottom-mounted ADCP and a culvert to collect water level and flow data. And just west of the Highway 1 bridge, there's a floating platform that captures other parameters such as temperature, nitrate, DO, and salinity. Down here marks the Castroville Slough bioreactor, where there's a pump that measures the amount of treated runoff that exits the bioreactor. Up here in the northwest corner of the map, around 4.5 kilometers away from Moro, Cojo, is the Caspian Weather Station, or the local meteorological station, and it collects data needed to calculate different terms in a box models used in the study. And lastly, I use data from NOAA's tide gauge located at the Monterey Harbor, and it's used as a proxy for water level in Monterey, or Moss Landing Harbor. And here are some pictures from the targeted deployments, where on the left here showed the sensors that I use, a microcat CTD measuring conductivity, temperature and depth, and a mini dot sensor measuring dissolved oxygen and temperature. And we have it band clamped on a T-stake that was eventually stuck into a few feet into the mud in the slough. Here we have it actually deployed in a field with a surface marker to tell us where to find it. Here's a picture at the head of the slough taken before we're about to go out and retrieve a sensor. And lastly, we have a picture of our pristine canoe after returning from our fieldwork. Now, when constructing the salinity and DO budget, it's important to first calculate the volume budget, which eventually feeds into some of those other terms used in the budgets. So here we can look at each individual term that contributes to the overall volume change of the system, and the color dots next to each term correspond to data sets used calculating that term. And that legend is on the top left of this slide. So first term in a budget was volume loss or gain through evaporation or precipitation, advection at the mouth, and the tide gate here is to remind you that it exists and plays a large role in regulating this tidal exchange term. Next, we have runoff from Castroville Slough measured by the flow meter at the bioreactor. And lastly, we have any residual flow not accounted by any of the other terms in the budget. Here's the salt budget, which uses the volume budget to calculate some of those terms. And we start off with the contribution from evaporation and precipitation over the entire surface area of the slough. Next, we have advection of water at the mouth, which includes the salt flux calculated at the Moro Cojo monitoring station using the ADCP and CTD sensor. And this term is essentially a product of flow Q and salinity measured at the culvert. Next, we have runoff from both Castroville Slough and any other runoff accounted for in the volume budget. And lastly, we have a residual term, which serves as a measurement of air in the salt budget. The last box motto for DO is for DL, which includes similar forcing terms. So first, we have gas exchange at the surface of the slough, which I had to calculate a piston velocity that's dependent on the wind speed and diffusion of DO at the surface of the water. And this is essentially a function of how oversaturated or undersaturated the system is. So if the system is oversaturated, say from extremely high productivity, there's going to be a net negative flux representing DO leaving the system and entering the atmosphere. When the system is undersaturated with low levels of DO in the system, there's going to be a net positive flux where the atmosphere diffuses DO into the surface. Next is the tidal exchange term, where it transports either oxygenated water from the ocean into the estuary or deoxygenated waters out of the estuary. And this is found by calculating the DO flux from the monitoring station at the mouth. We also take into account fully saturated runoff from Castroville Slough and surface runoff entering Moro Cojo. And the final term accounts for all other biological processes that either contributes DO or removes DO from the system. And this can represent activity both in the water column and in the benthic layer. So like any other model, mine comes with a list of assumptions. First, I assumed a constant area size for the slough to kind of simplify the model and have volume solely be a function of water level in the slough. So this also let me define the system as a box rather than accounting for any other increase or decrease of area in response to the complex bathymetry. And the area size was calculated by using a set of ordinary differential equations that related flow rate to the water level during the dry seasons. And then I optimized the parameters to eventually find this area size that fit this response. I also used stage height measured at Moro monitoring station to represent total water volume in a slough. And I did this again because of the complex and inconsistent bathymetry. So it would have been extremely difficult to kind of interpolate water depths along a slough. Next, I assumed the Caspian weather station located in Elkhorn Slough to represent atmospheric conditions over the entire Moro Slough. And this is because this was the closest meteorological data set I could find that had complete data for the time periods that I was looking at. And for the salinity budget, the monthly transects only provided spatial data with 1 month gaps in between. And when considering the Moro monitoring station, it had continuous data but lacked a spatial component. So when calculating the system's average salinity for each time step, I ended up using the monthly transects average over each station step and average over all the sites in a slough that day to get a system average. And then to resolve that spatial gap, I linearly interpolated the values over time. And similar to the salinity budget, the DO budget lacked the same resolution, but instead, but using a linear interpolation for the transects would just introduce a daytime bias since the transects were done during the day. So it misses any diurnal fluctuations you observe if you say measured the system at nighttime. And to resolve this, I used the DO measured at Moro monitoring station to represent the system average DO, so it would reflect that diurnal variability. And lastly, the flow data I got from Castroville Slough bioreactor had irregular measurements of the total flow and the length of time that it was on. So instead of assuming the total flow was discharged at a single point in time, I linearly interpolated in between each time step in that data set and assumed that it was a constant flow in between each of those time intervals. Next, I addressed our second objective, and that was to look at metabolic behavior on a seasonal scale. And this is just to see if the system is net heterotrophic, like many other estuaries in the US, or if it deviates from that behavior. And to calculate net ecosystem metabolism, again, we take the difference between gross primary productivity in a system and ecosystem respiration. And this can be broken down further into what is called biological oxygen change during the daytime and nighttime. And this is solely a function of how much DO is produced or removed from the system during those times. And as you can see in the bottom equation, we remove the influence of air-water diffusion or gas exchange and any other influence from advection to isolate that DO changes solely based on biological processes. So first, we'll look at any seasonal and temporal patterns observed in the data sets. Next, we'll look at the targeted deployments and a subset of the Moro monitoring station data to look for fortnightly and diurnal patterns. Next, I'll show a result of the box models to look for any terms that may contribute to the overall change at the end of the season. And lastly, we'll look at the net ecosystem metabolism and also its broken down components. Now, with that in mind, we can start looking at seasonal patterns from the data set, starting with rainfall here on the top. This figure shows the precipitation observed at the Caspian weather station. And the shaded background shows the time periods that cover the wet seasons, and a white background show periods during the dry seasons. And we can clearly see that there's a pattern characteristic of Mediterranean climate estuaries with little to no rain during the dry seasons and high precipitation rates during the wet seasons. The next plot below shows water levels in Monterey Bay, which follows an oscillatory cycle, and that the signal we observed here is primarily dominated by the M2 tidal signal. The next plot in red shows the water level measured in Moro Cojo, which shows a much more muted tidal signal than Monterey Bay, where you can see ranges from almost 0 meters to a little over half a meter, compared to Monterey Bay's, which varies between under 0 meters to a little over 2 meters high. And the reason why it's muted in the slough is, number one, due to the tide gate, and number two, due to the fact that the ADCP is placed at the bottom of the culvert. So the sensor's measurements of stage height is limited by that height of the culvert. And while water levels do go past that height from time to time, I just chose the water levels recorded by the ADCP for my data set just for consistency. The next figure in green shows PAR, or photosynthetically active radiation, which represents the amount of sunlight available for photosynthesis. And here we see a pattern where it reaches an annual maximum around July and a minimum around January. And the last plot below shows salinity measured at the mouth of the slough. I put red dashed line here to mark where salinity reaches 33 salinity units, and use this as a threshold for hypersalinity. So for any value that goes above that level, I consider it hypersaline. We can see that salinity stays mostly beneath this threshold for the wet seasons, mostly due to the higher precipitation rates, and that threshold gets crossed whenever we move into the dry seasons. The slide here shows a combination of both seasonal and spatial patterns for salinity, DO, and nitrate. The yellow lines plotted represent values measured at the Moro monitoring station, which is located at the mouth of the slough. And the color dots represent in situ measurements collected on the monthly transects. Starting with the top plot, we see salinity plotted as a function of time. And again, it shows the same seasonal pattern seen in the previous slide. But here we also see a spatial pattern as well from the colored dots. So looking at the wet seasons, we see typically lower salinity, but also the salinity for each site is grouped closer together. Compared that with the dry seasons, there's a much larger difference in salinity between the sites. And this is due to extremely high salinity values in upstream locations, which are more of the red hues dots on the plot. Next, I looked at DO, which show higher concentrations and higher spatial variability during the wet seasons compared to the dry season, where we see much lower concentrations and not as large of a spatial difference between the sites. And this is mostly likely due to higher productivity in the winter and springtime in the slough, which drive up the DO concentrations during the day. And much of those patterns can be explained by nitrate concentrations in the third panel, where we see much higher nitrate levels during the wet seasons due to increased runoff. And again, this drives higher productivity in slough and results in increased DO concentrations during the season. These patterns can be further explained with these gradient plots, where I plotted precipitation on the top panel to show the contrast between the wet and dry seasons. And the axis on the left for the gradient plot show distance from the mouth. So at the bottom of the axis shows 0 meters from the mouth. And as you move up along the y-axis, you're moving further upstream until you reach the very last transect site. And each X on a plot shows a sampling site. So I interpolated in between each site spatially and temporally to look for any trends that may exist. Now, starting with the second panel, it shows salinity. And again, we see seasonal patterns with higher salinity during the dry seasons and a larger spatial gradient, with the upstream areas reaching up to 90 units compared to the mid-30s at the mouth. Looking at the wet seasons, we see more of a mixed system where there's not as big of a difference between the head of the slough and the mouth. And this is due to more precipitation, which ends up freshening the slough and mixes the slough much more than during the dry seasons. Now, moving on to the last panel, which shows nitrate, it's a little harder to see any of the gradients, but we do see hotspots reaching up to 270 micromolar, show up around the middle of the estuary. And looking at the map on the left, these correspond to sites between 4 and 8, which are circled here. Showing that much of the nitrate spikes that are observed just upstream and downstream of where the Castroville Slough runoff enters Moro Cojo. So depending on the direction of the flow, it can either transport these nitrate-rich waters upstream or downstream at this point. So just to summarize, we saw that during the wet seasons, the increase in precipitation drives higher waters in Moro Cojo, lower salinity or freshening of the slough, higher nitrate levels in a form of hot spots around Castroville Slough outflow, and higher DO levels from increased productivity. And during the dry seasons, we see that tidal exchange is limited due to lower water levels in a slough, higher salinity, especially upstream where it reaches hypersaline values, lower nitrate concentrations, and lower DO from decreased productivity. So even though I did 3 deployments, I thought it'd be most worthwhile to go over the second deployment where I saw a noticeable trend in DO for sites T2 and T3. This particular deployment took place between February 4th and May 20th, so during the end of the wet season. And here on the top panel, we see some precipitation throughout this period. On the second panel in blue, we see the fortnightly cycle of tides in Monterey Bay. And although we don't see any noticeable fortnightly patterns in the DO panel underneath, we do see diurnal variability with the high happening during the day and low happening at nighttime. So looking closer at the DO for this time period, we see a steady decline in DO saturation percentage towards the end of the plot. And this steady decline can be attributed to all of that heightened productivity during the wet season finally subsiding. But the slough is then left with all this organic matter that's readily available to be consumed. So this then depletes the oxygen to sometimes hypoxic levels where I marked as any percentage under this dotted black line at 30%. And after calculating the amount of times hypoxic levels were reached for each site, T2 was hypoxic 7% at the time, versus T3 that was hypoxic 12% of the times, where the bulk of this occurred during the latter part of the wet season. And one reason for this was due to the location of the 2 sites. So T3 is actually located just west of where Castroville Slough meets Moro Cojo and is also shallower than the downstream T2 site. So because it's shallower and also has a higher likelihood of nutrient loading from Castroville Slough, it's more likely there is a higher productivity at the site, which lets it accumulate more of that organic matter to be fixed by organisms, thus increasing nighttime respiration and then depleting those oxygen levels even more. Lastly, it's easy to mistake the DO variability as following the fortnightly tidal cycle or the diurnal tidal cycle, but investigating the peaks further is clearly dictated by diurnal variability. So next, I looked a little closer at tidal and fortnightly variability during a 21-day subset of data collected at Moro Cojo monitoring station to look for any patterns or relationships between the data. I isolated the data to between July 1st and 21st in 2019, which falls into our definition of a dry season. And here we see a clear relationship between water level in the slough and flow, where whenever lower low water is reached during a tidal cycle, we see the black line, which shows water level in Monterey Bay drop. And when that water level drops beneath the blue line, which stands for water level in Moro Cojo, we see a discharge of water out of the slough shown in those dropping spikes in red on the second panel. Now, whenever the water level in black is higher than a blue line, we see either no flow or some flow into the slough. Now, comparing the flow with salinity on this panel, we see flow in red on the top panel and salinity in blue on the lower panel, along with the integrated salinity flux in a dotted black line with corresponding to the secondary y-axis. And here we see that during spring tides, we see a larger variability in salinity, and during neap tides, we see that the signals are muted a bit more. Next, looking at the salinity levels in relation to the Moro Cojo water level in green, we see that it reflects the diurnal tidal signal where when water levels are rising, water is flowing into the slough and pushes actually less saline water in. And during the fall of the tide, water exits the slough and transports more salty water out of the slough. And this inflow of fresher water tells us that even though there's no precipitation or very little during this period, since it is a dry season, there's still runoff coming from somewhere that eventually gets evected into the slough during this time. And we see a net loss of salt in the system over a 2-week period by looking at the integrated salinity flux over time, shown by a black dotted line, where it starts off at near zero, looking at the secondary y-axis. And after 14 days, ends up being at a value slightly lower, telling us that there was a net loss of salinity at this time or throughout this time. So even though the source of fresh water is unknown, it's very likely it could be derived from irrigation runoff from a nearby watershed. This next panel shows the same relationship, but for nitrate. where we see that during the neap tides, there's a rise in nitrate concentration, but during the spring tides, we see those levels decrease. Next, looking at the diurnal pattern, nitrate levels reflect the rise and fall of water levels in a slough, where high nitrate levels are detected on the inflow and lower levels are detected during the outflow. And this could be due to, again, freshwater runoff nearby entering the slough. So during the spring tides, we see a decrease in these levels, most likely due to stronger advection containing more ocean water than nearby freshwater runoff. Because flow is weaker during the neap tides, it's more likely that water from adjacent sources will be advected into the slough and less mixed with the ocean water. And looking at the integrated nitrate flux over time, we see that the system has a net gain of nitrate throughout this time period. And lastly, thinking about the relationship between this influx of fresh water associated with high nutrient levels, which are actually much higher than typical surface nitrate levels in Monterey Bay, which are roughly 5 micromolar, and nitrate-rich waters in the ocean or in Monterey Bay range from 30 to 40 micromolars. So all of this points to agricultural runoff from a different watershed as a source for this water. Next, I looked at the relationship between spring tides and neap tides and net ecosystem metabolism rates. So looking at the top panel, we see a clear neap and spring cycle happening, and compared it with net ecosystem metabolism rates in purple on the bottom panel, where we see that this rate alternates between net autotrophic and net heterotrophic, depending on which tidal cycle you're on. So during a spring tide where the tidal amplitudes are much higher, we see that the net ecosystem metabolism rates are net autotrophic and have higher primary productivity. And during the neap tides, we see net heterotrophic rates or higher ecosystem respiration. But in reality, when looking at the integrated net ecosystem metabolism for the system, we actually see a strong net heterotrophic system during spring tides that only weaken during the neap tides, but never reverse sign. So this shows us a little about the overall behavior over a fortnightly cycle during the dry season. Now, motivated by the nitrate relationship with the flow, I integrated the nitrate fluxes over time for the 2018 wet season and 2019 dry season to see if there are any differences between the two. And here on the left, we see that Moro Cojo clearly acts as a source or a net exporter of nitrate during the wet season, compared to the net import of nitrate in the dry season on the right, which makes Moro Cojo a sink during this time. Now, some takeaways for looking at the tidal and fortnightly variability. First, we see that flow and salinity modulates with the tidal cycle, and this depends on whether the water level in Moro Cojo is below or above the water level in Moss Landing Harbor. Second, we saw that nitrate variability depends on season and flow. So we see that high nitrate values enter the slough and low values exit the slough. And the magnitude depends on whether it's a spring tide or a neap tide. Next, we saw that DO is modulated primarily by diurnal factors, such as PAR availability, where higher PAR during the daytime drives high primary productivity, and lower PAR at nighttime allows for higher rates of respiration. And lastly, net ecosystem metabolism remains net heterotrophic during a portion of the dry season and only weakens during the neap tides. So moving on to budgets, we start with the volume budget first, where we integrated each term over the season and looked at the dominant terms, with this plot on the screen being for the 2018 wet season. And here we see that due to the high rates of precipitation, runoff in green dominates and contributes to the majority of water in the system. And this is then balanced by advection in orange that transports water out of the slough whenever water levels are higher than Moss Landing harbors. Looking at all of the other terms, we see in red that the total volume increases at the end of the season, and the atmospheric term in blue stays positive, which contributes water to the slough via precipitation. And looking at the dry season, which isn't pictured here, but I'll mention briefly, the dominant terms were evaporation and runoff from Castroville Slough, but the magnitude of these terms were a lot smaller than these terms seen here in the wet seasons. And similar to the volume budget, we integrated each term in a salinity budget to see their effects, where negative values show that that salt was removed from the system, and positive value means that that term adds salt into the system. Just as a reminder, 2018 was the wettest season in my data set. So for this season, we clearly see that we see the dominant terms are runoff and advection. And just like the volume budget, the runoff affects the slough in a large way throughout the season, and is then balanced by salt advected into the slough to retain that balance. And when considering the atmospheric term in cyan here, it is much less significant than of the other terms due to the fact that this estimates water entering and leaving the slough at the surface and doesn't account for any other surface runoff. The black line shows the error associated with the budget itself. And overall, we see a decrease in salinity storage in red for the 2018 wet season. So here I show the time integrated terms and took a weighted average for each season so we can weigh each season a little more evenly. Now we see a clear difference in magnitude for runoff for each season, with rain being measured at 33 centimeters in 2017 wet season and 57 centimeters in the 2018 wet season. And even though there's not much rain during the dry seasons, there's still some runoff. Those terms are still a little large because of the runoff from the Cojo Slough bioreactor. Next, looking at the atmospheric term, we see that even though this term is positive for all seasons, it's greater in the dry seasons due to higher evaporation rates. Next, looking at the advection term, this clearly dominates in the 2018 wet season, and is also positive for the 2017 wet season. Now, during the dry season, this term actually removes salt from the system instead. And lastly, we see that the system has a net gain of salt during the dry seasons and a total loss of salt during the wet seasons. Now, doing the same with the DL budget, I'll show the terms for both the 2018 dry and wet season here. And we can clearly see that the dry season is dominated and balanced by biological processes in black and gas exchange terms in cyan. Runoff and advection then add a small amount to the budget, whereas during the wet season, there's a mix of biological processes, advection, and runoff dominating the system. So looking at the weighted averages for DO, we see that advection removes DO from the system during the wet seasons and adds DO during the dry seasons. Biological processes add more DO to the system through higher productivity during these times, with a slightly smaller contribution in the 2018 dry season and even removal of DO during the 2019 dry season. Next, we see that the gas exchange term balances the biological processes term in a wet season and removes DO from the system in all the seasons. We see that runoff adds DO to the system with the highest contribution during the wet seasons where runoff is the highest. And overall, we see that there's a net loss of DO from the system during the wet seasons and again in the 2018 dry season. And looking at the results of the 2019 dry season, it's a little more difficult to conclude any of these terms since it had significantly fewer samples compared to all of the other seasons. So some takeaways from the box models. First, we see that high precipitation rates drive high surface runoff, which adds volume to the slough and is removed by advection to balance the system. The precipitation events drive strong runoff, which then freshen the slough and then have to be replenished through advection in order to balance the system. And we saw a net gain of salt during the dry seasons due to high evaporation rates and a net loss of salt in the wet seasons due to the rain. And lastly, DO in the system is dominated by biological processes and gas exchange during these dry seasons. And this is mostly driven by increased DO levels from high productivity, making the system super saturated, which then has to be removed by air-water diffusion, transporting that DO to the atmosphere as a result. And looking at the wet seasons, because of the increased precipitation, Advection and runoff then dominate during this time. Next, looking at the patterns for productivity and respiration, we see all 4 seasons here with the dry seasons on the left and wet seasons on the right, all integrated over time. And we see a clear difference in magnitude between the gross primary productivity rates in green between the wet and dry season, and also a clear distinction between respiration rates between the two. And this is mainly due to the increase in productivity during the wet seasons, which drive higher primary productivity rates and also lead to higher eco-respiration rates as well. And looking at the difference between these 2 terms, we get the net ecosystem metabolism, which we integrated in these plots. During the wet seasons, we can characterize the system as net autotrophic, as primary productivity dominates and results in a net positive net ecosystem metabolism at the end of the season, and net heterotrophic as values become negative or can be classified as nearly balanced if the net ecosystem metabolism is near 0. And one interesting thing to point out is in the beginning of the 2018 dry season, on the top right, we see that the net ecosystem metabolism actually increases during the first half of the season and then reverses mid-season. And this may be due to the fact that there was still precipitation and runoff during the first half of the season, which drives an increase in productivity. But after all of that gets shut down, the system reverses into a more heterotrophic system, which can then be seen happening early on in the 2019 dry season. And this just shows that even though we separated the seasons into wet and dry seasons, it's not always a accurate representation of the seasonal cycle, and this can vary from year to year. Now to summarize our findings for seasonal metabolic behavior, we categorize Moro Cojo as net autotrophic during the wet seasons due to higher precipitation, which leads to increased runoff. And that runoff drives high nutrient loading into the system and drives high productivity within the slough, creating a more autotrophic system. And one observation seen amongst many estuaries, especially those that are tidally restricted, is a surge in algal mats during the winter time and springtime. And in this case, and seen in a previous study by Mo-Wise, these algal mats dominate during springtide and can contribute to the increase in gross primary productivity. The tide gate also exacerbates this by not allowing normal tidal exchange to flush out these algal mats, which then allow these mats to proliferate throughout the springtime and then end up trapping all this organic matter within a slough. Next, we saw that Moro Cojo was net heterotrophic or nearly balanced during the dry seasons, and this is due to the limited flushing by both tide gate and lack of precipitation. And this leads to hypersaline waters upstream, limited export of organic carbon from the estuary, and more frequent hypoxic observations. Next, I did a comparison of Moro Cojo and 42 other estuaries, which were discussed in a synthesis study by Caffrey et al. I found Moro Cojo to be net autotrophic on an annual scale compared to the majority of other estuaries in Caffrey's study to be net heterotrophic. There was also lower primary productivity in summertime in Moro Cojo compared to higher productivity rates in the summertime for all of the other estuaries. I also did a closer comparison of Moro Cojo with Azevedo Pond, which is in Elkhorn Slough, which is a tidally restricted site in the synthesis study. But difference is that it had an area size 240% larger than Moro Cojo. But when scaling down the area size in relation to the net ecosystem metabolism rates, it was still found to be net heterotrophic. And even though it was tidally restricted, the main difference may have been due to the difference in nitrate loading concentrations, where Moro Cojo monitoring station detected a max nitrate value of 578 micromolar, and during the Caffrey study period, a max nitrate concentration of 130 micromolar was detected. So this large difference in nutrient availability may be a big reason why they both have these different metabolic behaviors. I also looked at comparing Moro Cojo with the South Marsh site in Caffrey's study to see if area size was a factor, since that estuary's area size was similar to Moro Cojo. But overall, it was found to be net heterotrophic despite it being in Elkhorn Slough and still prone to large nitrate loading. One interesting thing I found was that the few sites in Caffrey's study that were net autotrophic were categorized as submerged aquatic vegetation habitats. And in these habitats, it's usually dominated by seagrass and algal mats, which algal mats were also found in abundance in Moro Cojo during the wet seasons. So with this, Moro Cojo can be categorized as one of these submerged aquatic vegetation habitats and match the autotrophic behavior found in those systems in Caffrey's study. Next, I'll go over a sensitivity analysis I did for the box models, where first I changed the area size for all the seasons and found that some of the terms that relied on area size did change in magnitude, but in the end, the sign remained the same as if they contributed salinity or DO2 to the system, they still did after changing the area size, but they just had a different magnitude. Next, thinking about the net ecosystem metabolism, even though those calculations didn't depend on area size, the system during the dry seasons may become more heterotrophic when reducing the area size, mainly due to decreased nutrient loading and higher ecosystem respiration rates. Also, during the dry seasons, it's a little more difficult to predict whether the area size change would affect the behavior because the integrated net ecosystem metabolism for both 2018 and 2019 dry seasons were near zero. So any changes in area size can potentially push it over the edge to become net autotrophic instead. And lastly, in my model, the area size was estimated and fixed, but in reality, surface area is always changing in response to changing water levels, advection, or any other local forcings. And this could play a significant role or a factor in affecting some of these terms. But looking at a previous study done by Baker et al., their area size was different, but their estimated volume for the study site was similar to mine. So in the end, this gave me enough justification to use the area size that I calculated. So looking at the role of nitrate in Moro Cojo, we found that it acts as a source for nitrate during the wet season and a sink for nitrate in the dry season. And this can be seen in a spike in previous plots of the high nitrate inflow during the flood tide, and also the integrated nitrate flux plots, which show that the system supplies nitrate during the wet seasons and takes in nitrate during the dry seasons. And thinking about where this comes from, max concentrations in Moro Cojo were observed at 578 micromolar. Comparing this with values in the Old Salinas River that's just adjacent to Moro Cojo and also flows into Moss Landing Harbor, that's been measured to have an average nitrate concentration of 680 micromolar with max observations of 3,000 micromolars. So this leads us to believe that the nitrate stored in the slough may be sourced from Old Salinas River at the mouth instead of, say, surface runoff upstream in Moro Cojo. And just thinking about managerial implications for tidally restricted estuaries, this specific estuary deviates from the classic metabolic behavior found in over 90% of other US estuaries. And this is mainly due to the tide gate at the mouth, which limits flushing, retains organic matter, and shifts the metabolic behavior during the wet seasons. And this results in hypersaline and nitrate-rich waters that leave the system prone to eutrophication, hypoxic waters, and impaired water quality. And this is evidenced by the continual failing grades given by the annual Elkhorn Slough report card. And with the tide gate at the mouth, this also exacerbates a lot of issues the slough is already facing, such as contributing to hypersaline and hypoxic waters upstream, wetland degradation, increased habitat loss, shift in species composition, restricting the flushing of organic matter, which then shifts the metabolic behavior and contributes to the net autotrophic behavior of Moro Cojo. And lastly, the tide gate restricts the transport of organic carbon between the ocean and the estuary. So in conclusion, not only were seasonal patterns observed in response to tides, atmospheric forcing, and precipitation events, but we also saw patterns on a fortnightly and diurnal scale for salinity, nitrate, and DL. And through the box models, we saw that the main contributions to regulating salinity in the system was runoff and advection. For nitrate, it was advection of nitrate-rich waters into the slough and lower nitrate waters out of the slough. And lastly, DL was regulated mostly by the balance between biological processes and gas exchange. Looking at our second question, I was able to characterize Moro Cojo's metabolic behavior on a seasonal scale by using net ecosystem metabolism as a metric and considering the factors that would influence this behavior, such as the seasonal circulation, patterns of this estuary, tidal restrictions at the mouth, and excessive nitrate loading during the winter times. I was able to characterize the system as net autotrophic during the wet seasons and net heterotrophic or narrowly balanced during the dry seasons. Now, this analysis in this thesis and a box models can be used as a baseline for future work, such as monitoring shifts in metabolic behavior in response to variability of nitrate loading, different atmospheric conditions, or any additional hydrological modifications in the future. And we can also use the results to drive any managerial decisions related to preserving upstream habitats or mitigating saltwater inflow in the future. Now, before I end, I want to acknowledge the individuals, research groups, agencies, and institutions that played a hand in this research. Tom, I'm honored to have been advised by you and learned from you the past few years. Can't thank you enough for teaching me to become a better scientist and for constantly challenging me in the classroom, in the field, and in my research. You introduced me to the world of estuarine science, and I'm excited to continue research in this field, and even if it doesn't involve crawling around in waders and getting muddy. Next, Max, thank you for being such a great professor and committee member. Even though I took your class during that first COVID semester, I learned so much, and your class really opened my eyes to thinking about all the underlying processes that estuarine dynamics can affect and how it plays a large role in affecting our ecosystems. Next, Nick, thank you for being such a great committee member and for steering me in the right direction with all the literature you sent over. Your past work on the physics and its relationship to ecosystem metabolism really motivated my project and helped me think about how those relationships would or should behave in a system like Moro Cojo. Next, I want to thank the Central Coast Wetlands Group. Ross and Kevin specifically, thank you for letting me be part of the team and letting me present my work on behalf of CCWG. Your support these past years meant a lot to me, and I really appreciate you fielding any questions or concerns that I had related to the slough. And especially thank you for letting me borrow the canoe to deploy all of my equipment. Thank you to Jason Adelars and Steve Cunningham for your successful efforts in installing and maintaining all of the instrumentation throughout the slough. And thank you to the field team, especially for going out every single month and collecting the transect data, because after going out there a few times, it's really, really hard work. Next, I wanted to thank the Moro Cojo Slough Technical Advisory Committee for allowing me to be part of your meetings and deliberations on planning for the future of the slough. It's been extremely useful to see how scientists from different agencies, institutions, can come together and share their perspectives on the best way to manage this estuary. I also want to thank the entire Moss Landing Marine Lab staff, faculty, and community for being so welcoming, supportive, and encouraging during my time here. Thank you, Kieran, specifically for being an awesome teammate, paddling out in the slough and getting all muddy with me. Thank you, Marine Ops family, for letting me be part of the team and working alongside you these past few years. Gained so many valuable skills from you all, and I still think to this day that it's one of the best jobs out there, especially for a student. And lastly, I want to thank the Ocean Protection Council and the Coastal Conservation and Research for funding this project. And with that, I'm happy to take any questions.
Hey, thank you, Ryan. Great job. I think what we'll do is first see if there are any questions from the live audience, and then if there are any questions on Zoom, we can allow you to unmute and ask those questions. Any questions in the live audience? Ross, let me bring over the microphone. So Ross has a question here.
I have about 600 questions, Ryan, but I'll save those for later. Wonderful work, phenomenal set of data analysis. So thank you for doing this. It's going to greatly help our efforts to manage this estuary. And I'm going to have to watch your presentation like 4 more times to fully understand all the intricacies of your data analysis. I look forward to that. But quick question about the flushing and your box models, nutrients and carbon coming into the system, being metabolized, and then being discharged in some ways out of the system through the tide gates. Could you tell me again, you suggested that Moro Slough is a nitrogen fixer. Does that mean that we're accumulating nitrogen in the system and it is either being metabolized and emitted as nitrogen gas, or is actually being sequestered in the sediments? And the same question goes for carbon.
Yeah, that's a good point. I can't say for sure whether it is being converted to nitrogen gas, but that could be likely because as we see in the plots, that influx of nitrate entering a slough, that kind of remains in a slough, especially during the outflow. We don't see that same percentage or that same level concentration of nitrate exiting a slough. So a lot of that can end up being stored or fixed in a slough, either by all those organisms or just being fixed into nitrogen gas. And then same for carbon itself. A lot of that's just retained in the slough because, say, those ovomats, when they die, a lot of that doesn't necessarily get advected out of the slough, even though the tide gate is open at that time. There's still a kind of muted tidal exchange because of that. So a lot of that ovomats do end up being stored in a slough, and that carbon just kind of ends up sinking into the sediment and being stored there.
Thank you.
Any other questions?
Sure. Hey, Ryan, good talk. I like going to these thesis defenses. Thank you.
Thanks.
Hey, I got a question. So based on your research, are they going to fix that floodgate?
We're working on it.
I think that's the intention, to fix a floodgate. During the past couple of years, there was some deliberation with the Moro Slough Technical Advisory Committee on whether to add some orifices or small openings to allow for some exchange flow in between the ocean and estuary. That way, say in the summertime, all of that water, salty water in a slough, isn't just remaining stagnant and evaporating, which leads to like more hypersaline environments and hypoxic conditions in a slough. So I think the intention is to eventually fix that tide gate. The question right now is how much flow do they want to allow in between the estuary and the ocean to kind of keep it as a healthy ecosystem.
Yeah, because it doesn't look too healthy right now.
Yeah,
at least when I drive over it. So
yeah, especially during the springtime. I'm
Yeah.
sure you see it just covered in green. It doesn't even look like you can see some water underneath it.
well, hopefully your report will be the catalyst to get someone to make a move, huh?
Yeah, we'll see.
Yeah, yeah. Well, good job. Good job on your thesis
Thanks.
defense.
I have a question, just thinking about the tide gate and the management of the tide gate. So there are plans to repair and potentially adjust the flow at the tide gate. And you pulled together a lot of different data sources for your thesis. If you were to add one more component to that observing program, what do you think would help us best characterize the health of the ecosystem if we could add one more thing?
I think one additional, I guess, station that would help us characterize it would be a monitoring station up in a slough, because a lot of my work in those box models that I calculated, especially the DL box model, uses the DL measured at Moro Cojo to represent the entire slough, because I couldn't resolve that temporal resolution that the transects couldn't. So by having a continuous monitoring station up in a slough measuring DO, we can compare the net ecosystem metabolism at that point and see how it differs to, say, the mouth of the slough. And if there's a large difference, that means that there very well could be a bias in kind of using the monitoring station at the mouth to characterize the slough as a whole. So having that additional monitoring station up in a slough can give us that additional measurement and kind of serve as validation to see if that the deal at the mouth or the ecosystem behavior at the mouth of the slough is representative of the slough, of the entire slough.
We'll do it.
Okay, any other questions either in the room or online? Okay, I don't see any raised hands online, so let's in that case give Ryan another round of applause. Congratulations.
Thank you, everyone.
Thank you very much, everybody.
Great job, Ryan.
Thank you.